first of all, I want to start by saying two things. That one is that the title is perhaps somewhat flamboyant. I don't know exactly if it fits talk exactly well, but you can give me some feedback at the end. And number two is perhaps it doesn't classify so neatly under the genre of neuropsychology, but I hope I get my point across and tell together an all right story. So my objectives, put in an ambiguous sense, is to share my ideas about cellular and molecular neuroscience and the importance of it to investigating psychedelics, but particularly to also prompt discussion on the value of this sort of reductionist approach, so to say, to understanding, learning and memory. You could say, using invertebrates or simpler systems. It's not that invertebrates are simpler. Obviously, there are invertebrates that are, very complex, but some of them possess properties that are good for experimental work and they have been used, among other reasons, but they've been used historically for some important experiments. I'm going to try and sort of illustrate my points initially and some perspectives by just crisscrossing the talk with some quotes from some famous speakers, famous scientists. So to begin with, I want to sort of caricature the bottom-up approach, so to say, by quoting Francis Crick from this book, What Mad Pursuit. I recommend it. It's quite a nice little, small book. He says, so we also now appreciate that molecular biology is not a trivial aspect of biological systems. It is at the heart of the matter, and almost all aspects of life are engineered at the molecular level. And without understanding molecules, we can only have a very sketchy understanding of life itself. All approaches at a higher level are suspect until confirmed at the molecular level. And to illustrate this, there's just a schematic diagram of the double helix from the seminal paper in 1953 with Watson and Crick. I recommend reading it. It's one A4 page. It's open access. You can find it on the internet. And it gives a molecular basis to a concept such as hereditary, and they allude to that in the paper. And obviously, that's where Francis Crick is coming from. And sort of contrast this is the top-down approach, which I'm sort of quoting John Maynard Smith, who's quite a famous evolutionary biologist who applied game theory to biological systems. And he says in this book, The Problems of Biology, to assert that there are phenomena at higher levels which are likely to be discovered only by people who work at those levels, even though they are entirely consistent with the known physics and chemistry and may later be accounted for in those terms. I decided to attach a picture of Drosophila, the fruit fly, to this to sort of illustrate that, he could be talking about the organismal level, perhaps, or even populations of fruit flies. Fruit flies are often used in biochemistry studies, and it's a common system, a model organism that's very, very, very useful and very powerful, especially in genetics. And then I wanted to basically continue with a quote from John Maynard Smith, who says, well, most problems, he's talking about biological problems, are best solved by starting at both ends and trying to meet in the middle. Now, of course, when it comes to something like neuroscience, when you're trying to reconcile perhaps, psychology with, biological systems that's, controlled by, molecules and genetics and things like that, things can get confusing, and there's perhaps it can just get confusing, and I thought this sort of suited, this quote, this waterfall illusion by M.C. Escher, where if you sort of follow it from both ends, things don't really seem to make sense. It just got me thinking this picture. So, just to reiterate, the bottom-up approach you could call, especially within neuroscience, chemistry, biochemistry, things like molecular genetics. Of course, I've just picked 3 examples, by no means an exhaustive list. The top-down approach, we can talk about populations, organisms, and psychology and these kind of levels. Now, in psychology, in neuroscience, integrating these two is always a problem, right? how do we bridge the gap, so to say? I mean, the idea is to look at cellular networks, right, and circuits. But then you also have a problem, which is, I mean, these are vastly complex, and how do you quantify them? Can you model them? For example, using powerful, and computational techniques. I mean, it's just a really complex question. How do you integrate the bottom-up approach with the top-down approach? I think that's a major problem currently, particularly as neuroscience is such an interdisciplinary field. There's so many people working on different aspects of the problem. To sort of illustrate this idea of integration, I wanted to quote another person who was Charles Sherrington, who worked a lot on reflexes, muscular reflexes. And so he says, to move things is all that mankind can do, and for such the sole executant is muscle, whether in whispering a syllable or in felling a forest. And yeah, a professor of mine pointed out this isn't true if you believe in telekinesis. I don't know, there might be another talk this weekend on telekinesis by somebody else, who knows. But it's an important aspect that, I mean, people often look at the brain and especially perhaps, they forget that it's connected to, a non-neural system, such as, muscle groups, etc., so forth. So now I wanted to move on to a mollusk. It's a rather majestic picture of a mollusk. This is Aplysia californica, a sea slug. I think it's also known as sea hare. It's found on the coastal lines of California. It's been used historically in learning and memory experiments for various reasons. One, I think the main reason is because it was easy to work with, especially when technology for doing things like electrophysiology was quite rudimentary. And so I'll follow this up with some more information, which is that why would you study Aplysia? It seems a bit weird to study big sea slug, so to say. Well, the thing is vertebrates, such as the rat or even the human, contain a vast number of neurons. So the rat brain contains approximately 200 million neurons, the human brain approximately 85 billion neurons. And in vertebrates such as Aplysia or Limnaea, which is a snail, that's also used in learning and memory research, looking at the cellular and molecular aspects, similar to Aplysia, they have around 20,000 neurons. And the honeybee approximately a million neurons. But of course, I mean, the number isn't just important, and there are other properties that make the system useful, especially for scientists, which is their size, how easy you can work with them, and their identity, for example, from animal to animal, from preparation to preparation, okay? In Aplysia, you can isolate the same cells from preparation to preparation and basically work through different basically experimental paradigms and titrate chemicals and in the system and basically study what goes on with the behavior. Now, the behavior I'm talking about is related to a reflex action that the animal uses in the wild, which is the gill withdrawal reflex. And basically, you may be surprised to know, I don't know, I'm sure that many of you have heard of it before, but this even a simple organism like Aplysia can undergo various forms of conditioning, such as with classical conditioning with Pavlov's dogs, where he rings the bell, he has the food, and he basically, the dogs, he matches the stimuli. The dogs will salivate eventually after a certain conditioning paradigm when he just rings the bell rather than presenting the food. And I mean, you don't need to know any of the anatomy of this animal, but I just want to show this is the Aplysia turned upside down, and it's basically you can use a stimulus such as a tactile stimulus on the gill, and it sort of it contracts this. You compare it with a reward or a tail shock to the animal, and it can make these associations. This can be short-term and long-term in terms of how much, in terms of how it associates and learns to, that a tail shock or that a stimulus is not harmful or rewarding. And there's differences in the cells and the circuits when this happens. And it's all been mapped out in Aplysia, like I said, which is a major advantage. And it's sort of analogous to the external stimuli that's used in the learning and memory experiments. But the main interest for me was when I was learning about this, I was told that basically a key synapse in this system, which is, they call it the simple systems approach, is a monoaminergic synapse using serotonin. And I mean, there's a plethora of work that really delves into this synapse, and using dissociated cell culture experiments and complementary to many other different experiments, looking at the intracellular cascades of what happens using different pharmacological tools. The system's been really explored, and you can sort of alter learning and memory. In many ways, I mean, some people can, so say, implant false memories into Aplysia and make it think that it's learned, it's learned a danger or a reward in its environment when it's not there. So I just wanted to elaborate a bit on that, which is that basically many influential experiments can be done on this system, and they've given rise to important principles, such as the idea of synaptic plasticity, which was worked out primarily by Eric Kandel and his colleagues, using this system, and it focused predominantly on presynaptic mechanisms, such as long-term facilitation and long-term depression. And here's a bit more information about it. This is Eric Kandel holding one of those Aplysias. They don't look very nice. They're not as nice as that picture. But so they got the Nobel Prize in the year 2000 for their discoveries concerning signal transduction in the nervous system. Now, Carlson and Greengard weren't studying Aplysia. They were looking at dopamine and I think Parkinson's and some other systems. But what I wanted to highlight, following on from this, is that Aplysia in this simple invertebrate system has been really important, and it is complementary to work done in vertebrates. And Timothy Bliss and Lomo and others working in human brain tissue, mammalian brain tissue such as the mouse, discovered LTP around the same time, which is a post-synaptic mechanism. But it's now known that a lot of these mechanisms, they're common mechanisms, they're conserved, and in some way or another there's a bit of mix and match in different kinds of parts of the circuit. So there's generally consensus that learning and memory processes involve synapses such as the monoaminergic synapses, and there's lots of intracellular processes that are important for a drug's effects and target. If you knock out some of these intracellular signaling cascades, basically, such as the cyclic AMP-dependent processes, you can have very specific defects in animals, and in terms of learning and memory. Now, many psychedelic compounds interact with these pathways, and I think it would be a useful tool to explore the cellular molecular basis of learning and memory, to use, for example, radioactive labeled compounds and things like that in this kind of simple system to explore what's going on a bit more. I just wanted to hark back to the title, so you don't, so you don't feel robbed. But this is sort of what I meant in a kind of flamboyant way. But the skeptical mantras were sort of alluding to a scientific skepticism, a sort of reductionist framework using cellular molecular biology. And I think lots of invertebrate neuroscience is neglected, is perhaps for good reason. I mean, there is a lot of confusion. For example, when you look at receptors, and just using the serotonin receptors as another example, they've always, either the nomenclature is a bit different. So you've got, you've got an Aplysia, there's different homology. It might only be like 60% homology with the human receptors, but they've got different names and all the different domains and structural properties of the receptors. People think about them differently, and so there's not much integration between literature on synaptic plasticity, which is thought to be the basis of learning and memory, with invertebrate and vertebrate work. And then the orchestration of psychedelic studies within neuroscience, I sort of meant this kind of fundamental, more on the bottom scale research, it's more kind of it's bottom-up research, but it's also looking at neural circuits and that kind of things, and it'll help sort of researching psychedelics. It will help reduce the stigma in the scientific field, I think. It will allow it to be a bit more accepted, and it's complementary to the top-down approaches that's going on. Looking at, for example, mental health and end-of-life anxiety, and all these sort of the work done in medical fields such as with psychiatry. And as I said there, it's important to traverse from the fringe into the mainstream. And to illustrate this, I put this number line up. I don't know why it just sprung to my head, but I sort of use the analogy that the imaginary axis in mathematics was kind of rejected, and people were a bit cautious to accept the imaginary axis in mathematics. People would just think of numbers on one sort of horizontal number line. It took some time, but now it's generally accepted among mathematicians, and it's this kind of, it's complementary and it makes sense and it tests a lot of the crucial theories, and it harks back to the fundamental aspects of biological systems, and I think it's important. So as a summary, I want to just say that reductionist neuroscience, it can reveal, as I said, the fundamental aspects, but this may not necessarily be related to learning and memory. I mean, neurons are just specialized eukaryotic cells after all, and many people forget this. They often think of the brain as, they just forget the brain is just cells, the highly differentiated, specialized cells. And I mean, following on from that, it may reveal processes. For example, if you're testing psychedelic compounds, many of them which exist in plants, and interact with things like molluscs, which are a massive family of terrestrial animals and marine animals, that it could, well, not necessarily related to the psychedelic experience, but these compounds might have some sort of biological aspect to them. We can learn about the health, not just in humans, but in invertebrates, such as the honeybees or things like that, which are under danger at the moment. And we know, things, I don't know, it's a bit controversial, but for example, the neonicotinoid pesticides and these kind of things that, it's a controversial topic, but perhaps, testing the molecular pharmacology in invertebrate systems such as Drosophila, the honeybee, molluscs such as Aplysia and Limnaea, we can learn about their biology as well, and it's definitely important for us to know about their biology. And then my final point is just sort of how are we supposed to understand complex neural systems like the human brain if we only have partial and incomplete accounts of simple circuits in animals such as Aplysia. And from what I've gathered and from speaking to researchers looking at molluscan systems such as pond snails and Aplysia, I think there seems to be a lot of neglect, and people turn a bit of a blind eye to those that work on invertebrate systems. So I just wanted to share that with you. But I do think that in terms of the cellular molecular studies, it will be incredibly important. Thank you for listening. Cheers. Any questions?